So the next speaker is Clement Chow. He's an assistant professor at the Department of Human Genetics at University of Utah. And he's going to tell us about genetic and environmental modulation of NGLY1 deficiency.
So I want to start by thanking HUD for inviting me and thanking this community. I'm a fairly new person to this community. I started my lab about 2.5 years ago, and a couple weeks into starting my lab, this guy, Matt Mike, comes to me, and he's like, "You work on our pathway, and we want you to work on our gene." And you know how convincing he can be and how forceful he can be sometimes. And so this has started us down a journey of working on NGLY1 amongst many of other projects we have in the lab. But today I want to tell you a little bit about some of the work we've been doing in the fly model of NGLY1 and how we've come across several different ways we think we can genetically or environmentally modulate the outcome of loss of this gene function in the fly. And after hearing several of these talks today, I realized that our work, all of our work really converges on similar themes and pathways, and I think you'll see that in my work as well. So my lab is broadly interested in understanding how genetic variation impacts disease outcomes and therapy, responses to therapies, and this is basically, everyone in this room knows about this. So if you have a group of patients, they're all different in how they manifest that disease, and they all are different in how they respond to therapy, and we really want to understand how the background genetics between those individuals impacts those disease outcomes. And this very simple diagram I've shown you really belies the fact that we really don't understand this at all, even though you know, we're pushing towards precision medicine, but we really don't understand at all how the background genetics and the genome that everyone has influences that primary disease mutation or problem. And so at the heart of this problem is trying to understand these two ideas of reduced penetrance and variable expressivity. So you have this kind of standard autosomal dominant pedigree where you have affected individuals that carry the disease and they pass it on, and 50% of the people in this family have the disease. But the real world problem is that we have unaffected carriers, and this is what we call reduced penetrance, people who carry the mutation but don't get sick. And we also have carriers with variable disease phenotypes, as again, we all know about this, where different patients, even within the same family, can manifest the disease quite differently. And of course, this is all a complex interaction between the genetics and the environment. And again, my lab is really interested in trying to understand how this interaction really gives rise to disease variation and the differences we see between patients. So this is really, you know, we're trying to understand the phenotypic spectrum in rare disease. And so we have this kind of really simple example of a disease phenotype or some kind of symptom in patients. And as we all know, some patients are on one side of the distribution and do a lot better than other patients on this distribution. And the way I've shown you this is really to indicate that these simple Mendelian diseases, these conditions where we know the causative mutation, the single gene that's mutated, are really quantitative traits. They really should be treated like cardiac disease or height, for example, where you have every phenotype in between the tallest and the shortest person. And this is what every Mendelian phenotype is like as well, even though we tend to simplify it in the lab and in textbooks. And of course, this kind of understanding of rare disease and diseases in general is important for diagnosis and therapy, and especially as we push towards precision medicine. Okay, so why rare disease? Of course, everyone in this group knows there's been a real rediscovery of rare disease in the human genetics world. For about, I think, 10 years, the human genetics world forgot that Mendelian diseases exist, and everyone was working on complex problems like cardiac disease and high cholesterol. But I think with new technologies, we've rediscovered that this is actually a problem for human health. And again, you know, as everyone knows here, patients are identified daily and patient groups are growing, which is really great for our work as well. And we have the technology and the model organism tools to really kind of push the limits on the way we ask questions about rare disease in the lab, and I think that that's made for really exciting science. Okay, so my lab wants to take what we learn from patients and in the clinic into model organisms, and we usually try to ask questions related to modifier genes for most of the projects we work on. But in some cases, we actually have to ask questions about pathogenesis for models like NGLY1, where we actually don't know a lot about the disease and don't know a lot about what's happening in the fly model. And so we kind of have to marry these two approaches to really understand the biology behind why patients might be so different from each other. And the hope, of course, is to eventually bring these findings back to the clinic and back to the patients. Okay, so I'm going to skip the slide about NGLY1 deficiency. We all know about this. But we went into this project with a really basic understanding of NGLY1 function. And my lab kind of likes to take this very kind of basic hypothesis-free approach of not to try to come up with the questions that we think are the right questions, but let the biology ask. So we had kind of this really basic understanding that NGLY1 has deglycosylation function. And this was just kind of based on some of the work that Tadashi had published and just kind of looking at the protein and what we thought it might be doing. And of course, based on previous findings, we thought there's probably a pretty good chance it's also involved in this ER-associated degradation, or ERAD. And so, as we've all seen, misfolded proteins can accumulate in the lumen of the ER. And these misfolded proteins are glycosylated, and then ERAD pulls them out from the ER to the cytoplasm, where NGLY1 comes and deglycosylates them. You've heard this. And then these misfolded proteins can go to the proteasome to be degraded. Okay, so this kind of really basic view of what NGLY1 we thought NGLY1 might be doing gave us some really obvious hypotheses to ask, to kind of formulate. So if NGLY1 was missing, we'd expect there to be ER stress in the cell. As misfolded proteins are not properly degraded through ERAD, they might be backed up into the ER and might cause ER stress. We also might get cytoplasmic aggregates in the cells, because if NGLY1 can't deglycosylate these proteins, as Tadashi has shown, they might accumulate in the cytoplasm and not be properly degraded. And we also might get some glycan recycling problems. And the nice thing about all three of these hypotheses is that they all give very obvious transcriptional outputs if we look at the transcriptional profile of the cells or the animals. So that's kind of what we started with. And I'm going to talk, all the work I'm telling you about was all done by a really talented technician, Katie Owings, who had the luck of working with just me for almost 9 months before someone else joined the lab. So she's now a graduate student in the lab, and she's quite talented. All right, so we set out to first develop a model, Drosophila model of NGLY1 deficiency, and we kind of wanted to just jump right into this. So we just went straight to the RNAi method of knocking down the gene. We've heard a little bit about this either yesterday and with similar methods that Gary has used. So we used RNAi to knock down NGLY1 transcript all over the fly, throughout the body and every tissue. This is just expression data showing that we essentially almost completely get rid of the expression of NGLY1 in these flies. And so, not surprisingly, loss of NGLY1 results in lethality in these flies. We get about 25% of these flies that survive to adulthood. So it's very similar to what Tadashi is talking about, that we have this dead fly and we don't know much what to do with it. So as I said, we like to kind of approach science with hypothesis-free approaches, and so we just let the science the biology tell us what question to ask next. So what we did was we performed RNA-seq analysis, which is a way to measure all the different RNA transcripts in the fly all at once, and look for different changes that might tell us about the biology. And so we did this, and we compared the NGLY1 deficient flies to our control flies, and the changes were actually quite modest. We only saw about 100 genes that were upregulated and about 300 genes that were downregulated. And the first thing you do when you do this kind of analysis is do something called a GO analysis, which tells you a lot about the genes that are enriched or what's in common with those genes. And so we did that for the upregulated genes, and what this showed us was that there was no evidence for ER stress in these flies. If there was ER stress, we'd see enrichment for ER stress response genes, and we saw none of that. In fact, all these GO enrichment categories are all just fairly general stress response genes that you see a lot of times in sick flies. So if you take any mutants and do these types of transcriptome analysis, you often see these responses, just because this is what happens in a fly when it's sick. We did see this response to heat, which I'm going to get back to later in the talk. But again, this really told us that loss of NGLY1 function on its own is not really sufficient to induce ER stress, at least in the fly and the way we kind of did this measurement. We've also looked at NGLY1 function in a number of models of ER stress in the fly, and it doesn't seem to affect the function of ER stress in any of those fly models as well. Okay, so we also did this Go analysis on the downregulated genes, and I'm not going to go into a lot of this, but we saw what confirmed the study that Gary just talked about, that the fly seems to have a defect in proteasome and oxidative reduction processes. These are both related to NRF1 function, and in fact, these over 15% of our downregulated genes are known to be regulated by NRF1 in the fly. And so this is kind of a nice cross-model organism confirmation of biology that seems to be converging on NGLY1. Okay, so I'm not going to talk about things that we already know about. What we next did was we just poured through the list of genes that seem to be up and down regulated to see what might be interesting. And one thing really caught Katie's eye, that the gene GFAT1 was down about 50% in the NGLY1 deficient flies. So what's GFAT1? In fact, GFAT1 is the rate-limiting step in the production of UDP GlcNAc from glucose. And so this is really converging on what Matt was finding with his son Bertrand. And so we found that GFAT1, so it's a rate-limiting step, and so if GFAT1 is down in these flies, we expect the final product to be down in the flies as well. So we expect that UDP GlcNAc is also decreased in these flies. And so UDP GlcNAc, of course, is involved in all kinds of proteostasis functions and in modulating different proteins. And you can see N-glycosylation, O-glycosylation, and a bunch of other functions. And we thought, well, the problem is that GlcNAc is downregulated in these flies, then all these processes must also be downregulated. And that might be contributing to the lethality we observe in these flies. And so Katie made the really simple hypothesis that why don't we just raise these levels in the fly, and if it's a problem, then it should raise these processes back to normal, and it should rescue the lethality. And so she did this really simple experiment where we raised the flies on our normal food, or food supplemented with GlcNAc, and we let them develop through their normal life cycle of larval development, pupation, and then we counted them and scored them as adults. So on the normal food with no supplementation, we get about 30% survival to adulthood after about 11 days of collection. This is actually a much longer developmental time point than you get in wild-type flies. But when we raised these flies on GlcNAc, what we saw was nearly 70% of the flies surviving to adulthood. And you can also see that they're coming out into adulthood much sooner than without GlcNAc. And this is really nice, as Matt has indicated, that a supplement like GlcNAc might be able to provide some therapeutic relief, at least in the fly model, that we can buy a supplement like this on Amazon or at GNC, and it's already being used for some other kinds of conditions. And this is, I think, really exciting for a rare disease where something so readily available might provide some form of relief. It's of course not a fix, because these flies that survive to adulthood are actually still quite sick. So it's not fixing them, but they're not dying anymore, which is an obvious move towards in the right direction. We also showed that when we also did RNA-seq on these flies that were raised on GlcNAc to look at their transcriptomes, and in fact we think that the GlcNAc rescues functions completely independent of the NRF1 defect, because we see the same exact defect in these flies that you'd expect from NRF1 problems, treated on GlcNAc and not treated with GlcNAc. So we think, again, like Tadashi said, this is an independent mechanism to rescuing some form of lethality in these flies. Okay, so Katie dug a little more in this RNA-seq data, and I mentioned earlier that we saw this kind of enrichment for response to heat, this heat shock response in the NGLY1 deficient flies. And this is just a number of the heat shock genes that we saw upregulated in these flies. And you can see that this is actually quite a modest heat shock response, with the exception of HSP70, which is upregulated about 7-fold. Lots of problems, lots of mutations in flies can cause a heat shock response in the transcriptome, and it's usually a much larger response than this. And so this is actually, again, quite modest. But of course, this made a little sense to us because if there were cytoplasmic aggregates and to a lesser extent ER stress occurring in the flies, we might expect a heat shock response, which is what this response is evolved for in the cell. And so Katie thought, well, this is kind of a weak response. What if we just hit the flies with a much stronger heat shock response? What if we just turn that dial up even more? What will happen to those flies? And so in flies, it turns out that there's actually a really nice protocol for doing heat shock in the fly. It's used for regulating a bunch of gene expression that's been studied for years. And so we just took those protocols and applied it to the NGLY1 deficient flies. So Katie applied heat shock to 3 different developmental time points during larval development, either in the first instar developmental stage, the second instar, or the third instar stage, 1 hour for 37 degrees. And then she allowed them to develop it normally, and then again, we counted them as adults. And again, with no treatment, we get about 30% survival to adulthood. But when we hit them with heat shock at these 3 different time points, we see that there's quite a substantial rescue again in the lethality. And we see the best rescue occurring during the first instar of larval development, suggesting that perhaps there's some process that's happening here that really benefits from having a strong heat shock response that might help push the fly through development. And so turning up a heat shock response in these flies may provide some rescue for this lethality. Katie went on to show that, in fact, these heat shock genes that were upregulated in the fly are actually required for survival in the fly. And so she went on to knock down each heat shock gene on its own on the background of these NGLY1 deficient flies. And what you see is that when we knock down HSP70 on its own in the NGLY1 background, we get 0 survival, telling us that the function of HSP70 is actually absolutely required for these flies to survive, even the 30% that survives to adulthood. We see a similar effect with the other heat shock genes that we knock down, but it's a smaller effect. But in every case, except for HSP68, there's a requirement for those heat shock genes for survival. And of course, as many other people have shown in the past, when you knock down heat shock genes on their own, they don't really have an effect on lethality. So this is a genetic interaction between NGLY1 and the heat shock pathway. And again, we think that this is independent of the NRF1 defect that Gary's group and a bunch of other groups have shown now. Okay, and again, this is exciting because the heat shock response is another one of these responses where molecules are being developed as drugs for a number of other kinds of diseases, like neurodegenerative diseases, and especially drugs are being developed to upregulate the function of HSP70. And here we clearly show that HSP70 is required for the survival of these flies. And so that's another kind of way we may be able to piggyback on existing efforts to develop drugs for NGLY1. Okay, so I want to just change topics just a little bit and kind of get back to the heart of what my lab is interested in, is why are patients so phenotypically variable across the patient group. And this is just from the 2014 paper, and we see that there are very few phenotypes amongst these NGLY1 deficient patients that are completely consistent across all the patients that have been characterized. And so this is really exciting to us that there might be genetic modifiers in the background of these patients that make some of them better off than others. And we really wanted to try to get a hold of this and try to model it in the lab. And so how do we do this? We use this resource in Drosophila genetics called the Drosophila Genetic Reference Panel, or DGRP. And this is a collection of wild-derived strains from a natural population of Raleigh, North Carolina. It turns out if you work with Drosophila, the natural population is just a farmer's market. And so a bunch of these strains were collected, and what's important is that they contain natural genetic variation. So this is variation that's consistent with life, that's floating around in the population. So it's a bit different from what you heard from Gary, where they induce the genetic variation to look for effects. We're looking for just effects of variants that are just floating around in the people in this room, for example, or in flies in a population. So we have about 200 strains in the DGRP. Each strain is homozygous and inbred and represents a single genome from that population. So you can kind of view them as 200 individuals. And most importantly, we have the whole genome sequence of all 200 strains, making it really easy for us to do phenotype-genotype studies quite quickly. And so what we're essentially doing for NGLY1 deficiency and a bunch of other diseases now is testing a disease model on 200 different genetic backgrounds, basically asking how are these 200 individuals differing in their phenotypes and what genetic variation drives that. So we take a disease model and we cross it into the DGRP, and then we measure some quantitative phenotype, and as I like to say, every phenotype is quantitative if you measure hard enough. And then we run an association analysis to try to identify modifier genes, and then we really dig into the function of those modifier genes and how they're affecting that particular disease model. So with NGLY1 deficiency, we took the fly model that I described earlier, we crossed it into the DGRP, and then we use lethality as our phenotype. Again, we still really have this kind of prominent effect of death during adulthood that we can use as our phenotype. And then we ran an association analysis to identify modifier genes. Now this is very much still kind of an ongoing project, so I'm just going to tell you about how far we've gotten and how promising it looks. So this is what the screen looks like. This is the proportion of surviving flies at the top of this. At the top of this distribution is no death, 0% lethality, and 100% lethality at the bottom of this distribution. This thing's not working anymore. All right. And so I have a backup laser pointer actually, so I'm going to use this one. So so at the bottom is 100% lethality, and so what you see on the left side, so these are the 200 DGRP strains, all with NGLY1 function missing from them. So they only differ by their genetic background. And what you see is that the majority of the strains on the left side of the distribution don't do very well without NGLY1 function, right? And that's expected from the model standard background we used earlier, and just from what we know from all the other studies. But what's really striking is that on this side of the distribution, there are a number of strains that essentially can live just fine without NGLY1 function at all. And so that's really, really striking, right? We weren't expecting to see any flies that would do this well. But there are some genetic backgrounds that seem to do okay without NGLY1 function at all. And so what we really want to understand is what genetic variants are these flies carrying that make them so much better at dealing with loss of this gene function versus these flies that can't even live into adulthood without this function. And so we ran that association analysis, and we pulled out a bunch of modifier genes, and I'm not gonna show you the whole list, but I'm gonna show you a cherry-picked list. But one thing that really stood out to us was that a bunch of these modifiers are involved in protein homeostasis, and you'll see functions here related. So this is Drosophila gene name, human gene name, and functions involved in ERAD, protein trafficking, a number of ER resident proteins, ubiquitin, ligases, end-link glycosylation. So we see that our modifiers are converging with a bunch of other biology you heard this morning. And you also, some other kind of interesting things to point out is this HRD3 cell1-like modifier was also a modifier pulled out in Gary's original screen for things that regulated NRF1. So that's interesting. And we also have a number of, so for example, this TMTC2 gene, we actually see both orthologs of the fly version of the human TMTC gene being pulled out, which suggests that is particularly important. And we also have both orthologs of the ERMP1 ER metalloprotease gene being pulled out, which again suggests that might be especially important. So again, multiple hits suggest that things are really, really interesting, as Gary pointed out. So I'm just going to highlight this HRD3 cell1-like ERAD protein for you. So what we found was a serine to proline change in cell 1-like that seems to modify the lethality of NGLY1 deficiency in the flies. And what we saw is that there was a modest gain in survival when the flies carried a proline at this position compared to flies carrying a serine. And so what is this gene doing? It turns out that cell 1-like is actually part of the ERAD complex that pulls misfolded proteins out of the ER into the cytoplasm. This serine to proline change is in the cytoplasmic tail, sitting right here. And we think NGLY1 probably functions somewhere kind of in this space in the cell. And so the physical location of NGLY1 protein and this serine to proline change in cell 1-like really suggests that there's some interesting biology, perhaps even a physical interaction between cell 1-like and NGLY1. So today I've shown you several ways we think we can modify NGLY1 deficiency. A diet-based method using GlcNAc, on kind of environmental manipulation using heat shock, and genetic modifiers that point to ERAD and other proteostasis-related functions. Of course, we're not really sure whether these are all related or independent mechanisms. We know that at least the heat shock and diet-based methods are probably independent of NRF1, but it's to be determined whether all these are kind of converging on the same pathway, or whether we're really finding independent ways that we should be targeting NGLY1 deficiency. So it's always important to remind you that all this work is brought to you by model organisms and the importance of model organism work. I know that this is kind of the choir, but it's important to remind people that model organism work is the only way we're going to make progress on rare diseases and thinking about how we move forward with therapies. And we want to know how we can help the clinician and patient. So my lab studies other rare diseases as well, and so if you have a disease or a gene that could benefit from the types of analysis that we do, we are happy to talk, and we are happy to kind of hear from patients as well, especially the NGLY1 community, about their experiences. That kind of information helps us to prioritize which genes and which pathways we want to go after first. So we are happy to talk, and I'm happy to talk afterwards. So I just want to thank my lab, especially Katie Owings, who did nearly all the work that I presented here. We work closely with NGLY1.org and Matt Might. A bunch of my funding here, and my lab is always recruiting, so if you're interested or you just want to chat about our work, please contact me. Thanks.
Thank you very much.
Very nice. So the heat shock and the GlcNAc, is that additive? Have you tried?
Sorry, what?
Heat shock and GlcNAc supplement, is that additive effect they have?
It's not additive. We don't get a benefit when we do the two together. I don't know what it means.
Okay, so then the other question is that the one that who has the 100% survive, that strain is the fertile or sterile? Because fertility has been the problem.
I don't know. They're surviving, but they're not healthy.
Oh, okay.
So they're not
still not healthy
flies. They look quite sick, but they are surviving, and so that's, I think.
In your heat shock experiments, which is a fairly non-specific hormetic response evoking procedure, have you tried to see whether all the different heat shock genes are expressed, and whether you could rescue your lethality by just overexpression of heat shock genes?
Right, individual heat shock genes, you mean?
Yeah.
We haven't done that experiment. That's mainly, the ones I showed were mainly the ones that were upregulated. Of course, by knockdown, we know that those are required for survival too, so I suspect if we upregulated them more, it would probably provide a benefit.
And do you get a modification of the phenotype by changes in diet, like in low caloric diets, for example, or high sugar diets? And
we haven't tried low caloric, but we have tried a bunch of other sugars, and nothing else seems to make an effect except for GlcNAc.
Okay, thank you.
A very interesting talk. I have a common question about your gene modifier. How do you define them? They have higher expression level, or they are just more functional because they have some mutations in those genes?
Well, so this is a GWAS, and so we're using just genetic association. So we don't know what the SNPs are doing to that modifier.
Those are SNPs.
Yeah.
Okay.
So this is we ask which SNPs are associated with the lethality that we see, and so that tags a gene. And then we go after the gene. We're not actually sure right now how all these modifier genes are having their effect, with the exception of the HRD3 cell 1, like where we have a serine to protein change. I mean, I think a protein change is pretty clearly going to be problematic for the protein.
Okay, so you're not very sure whether they are more functional to compensate the loss of NGLY1.
Well, the way a GWAS works is that flies that carry one allele have better function than flies that carry the other allele. So one group of flies has better function for that gene, and one group of flies has lower function. That's what we're asking in the analysis. Right. Yeah.
Got it. Thank you.
Yeah.
So in that SEER to, what is it, SEER to P?
Yeah.
Yeah. What does that effect have on the activity of the cell 1?
We really want to know. I think that's what we're going after next. No one studied the cytoplasmic tails. You probably know of this gene. Everyone cares about the business end. But there's about 50 or 60 amino acids sticking out into the cytoplasm that no one's looked at. So it's really interesting. Yeah.
That's a great talk. So in your RNAi knockdown experiments, you identified a number of genes that are upregulated and downregulated that rescue lethality, basically. Have you looked at any gene expression databases to look for products that have already been approved that will offset that activity? For example, products that upregulate GFAT1.
Yeah, I think Andrew Su is supposed to help us with that. Is he in the room? Yeah. So we're talking a little bit with Andrew Su and trying to apply some of his more sophisticated methods in trying to do this. This is something we could obviously just pour through the databases and look, but I think that there's probably a little bit more sophisticated ways to do it than Andrew's probably going to help us with. Yeah. Thank you.